The first online revision science class covering the content for your combined science GCSE specification from AQA. So what we're going to focus on in this session is cell biology, the first unit of work you will have done during your GCSE course, looking at the basic units of all forms of life and exploring structural differences between different types of cells and how that enables them to form specific functions within organisms. So you'll have been provided on milk with a handout just like this, so if you want to make any notes on it while we go through the video, you're more than welcome to. And then there is also a follow-up educate quiz that goes with this video as well, that you can complete after going through it in order to jog your memory and see if you're able to apply any of the things that we look at in this video. So if we go on to our first section. So our first section is on eukaryotes and prokaryotes. So key difference to begin with, prokaryotes and eukaryotes. In terms of a key definition, you've got to remember that eukaryotic cells, we're often referring to plants and animals. So referring to plants and animal cells. And the fact is that they have a cell membrane, cytoplasm, and importantly, they have genetic material enclosed in a nucleus. Whereas with prokaryotic cells, we're often referring to bacterial cells. And they, importantly, have no membrane-bound organelles. So these are organelles that are contained within a membrane. Good. Now, importantly, you'll see here we've got some of the defining attributes of eukaryotic cells and prokaryotic cells. So when we're talking about eukaryotic cells, they have membrane-bound organelles. So we're talking about nuclei, we're talking about mitochondria, ribosomes. When in plants, they have a cellulose-based cell wall. So cellulose is what cell walls are made out of. You've got DNA on chromosomes, and DNA, importantly, is held within a nucleus. They have mitochondria, in fact, they have nucleus, they have ribosomes, and down here there's some extra information that is A-level, so don't worry too much about that. Now, the differences in terms of prokaryotes and bacteria, instead of having a nucleus, their genetic material is often held in a single free loop of DNA. And they also might have small rings of DNA called plasmids. So the key differences between eukaryotic cells and prokaryotic cells that you'd be expected to know. So have a look at the next recap. Now, on the next recap, you've got the differences between plants and animal cells. So when we are talking about organelles, we are talking about things that make up or contained within these cells, okay? So these are all organelles. So these are all found within cells, and they have specific functions to allow the cell to carry out its unique jobs. So you've got the nucleus, which both contain, which controls what the cell does. You've got the cytoplasm, where chemical reactions happen. You've got cell membrane, which holds together and controls What goes in and out of the cell? Mitochondria turns glucose and oxygen into energy. And if you remember, this is where it's the site of respiration. So it's where respiration occurs. Whereas the key thing to remember here is the differences between them. So what does a plant cell have that animal cells don't? They have a cell wall, which, as we've said, is made of cellulose to give support and also structure to the cell and subsequently to the plant. Vacuole, which is a storage area within the cell which contains cell sap, which has got sugar and salts in, which obviously plant cells can then use as required. And then they contain chloroplast, which contains chlorophyll, which is a green pigment. It's a colouring, and it's the site of photosynthesis. So it's where photosynthesis occurs. Okay, so it's important to remember that. And so it's important to remember that they both have these, but only plant cells have these. Okay. Good. So hopefully you made a few notes on that. And then if we have a look at what we call cell specialization. Now, cell specialization is when the structure, so cell specialization is when the structure of a cell, so it's when the structure of a cell relates to its specific function. So, for example, if you look at sperm cells, they carry genetic information. They do this for reproduction. And one of their adaptations is obviously they have tails to swim, so that they can meet eggs and fertilise. You've got red blood cells. Now, these are adapted to carry oxygen to cells away from the body. So you might say, for example, they have a large surface area to fit more oxygen in, and also they've got no nucleus, so it's got more space to fit oxygen in. Nerve cells, very long, to carry messages around the body. Also, they are insulated by something called a myelin sheath. And what this does, this improves the speed at which messages are communicated. A couple of other examples, you might have the root hair cell, which due to its shape has a big surface area so it can take in more water. And you've got palisade cells in the leaf. Now these contain lots of chloroplasts, so that you can increase the amount of photosynthesis. Just a couple of examples of specific cells that have specific shapes and things within them that allow them to carry out unique jobs. Now, when we're talking about microscopy, there are two main types of microscope you need to know a little bit about. Electron and light microscopes is the required practical that you'll review in school, looking at magnification, but there's an important formula to link to calculating magnification. So firstly, remember, you've got to realise that magnification is how big an image is compared to the original object. So you're looking at an object down a microscope and you have an image that is times 10 magnification, times 20 magnification. That means that the image is 10 times greater or 20 times greater than the actual object itself. Now, there's also the resolution of a microscope image. So when we're talking about the resolution, we're talking about the clarity. How clear the image is. Is it fuzzy or is it clear? Is it crisp? Can you see what it is? And resolution is actually the ability to differentiate between 2 points on an image. So can you tell 2 separate points apart, or actually are they blurred together? So if you can tell them apart, they've got high resolution. If they're blurred together, if it's fuzzy, if you can't tell what the image is, it means it's got a low resolution. Good. Now, the main thing is to be able to understand some of the differences between them. And so we've got differences here between comparing the light and electron microscope. So, magnification and resolution. So on these, my electron microscope has a much higher magnification and resolution, which means you can see it. So you can see objects clearer and in more detail. Now, disadvantages of these is you can only look at dead or abiotic samples. This is because in order to see them, you use electrons. So samples are exposed to radiation, which is why you can only use dead ones. They're also incredibly big, difficult to move, and also what you might describe as being quite expensive as well. Whereas light microscopes are much cheaper and easier to move. They're small and portable, and because you're only exposing a object, a sample to light, you can use living and moving organisms as well as dead ones. So those are key comparisons. So it's about learning the differences. Good, right, let's have a look at the next bit. So the next area to be having a look at, and remind yourself about, is cell division. So cells dividing to produce new ones. Now, bearing in mind about the structure of a cell and what is within the nucleus. So the nucleus of every cell contains chromosomes, made of DNA molecules. Now, these themselves, chromosomes, carry genes. And genes determine features of who or what an organism is. Now, we've got a main process of cell division you need to be aware of, and that is mitosis. Now, mitosis in itself, it's important for growth and development of an organism. It's important for growth and development of an organism. Now, the end product of mitosis is that you are producing two identical cells, okay? So it's a form of asexual reproduction, or it's almost cloning. So a cell is producing a clone of itself. So in your first step, you have your chromosomes, and then the chromosomes themselves duplicate. So they double. And then what they end up doing is they end up being pulled apart, the chromosomes, the double chromosomes, to make two identical sets which have the same DNA as the original cell. And then finally, two sets of DNA split into two cells. It's an important thing to remember about mitosis. It's for growth and repair, and what's produced is identical. So two cells are identical, and you just have one cell division. So the cell itself just divides once to produce this identical cloned cell. Now, this process can also play a role in what we refer to the use of stem cells. Now, a stem cell itself, in terms of if we were going to define it, it's an undifferentiated cell of an organism, and it's capable of producing cells of a specific function. So when we're talking about undifferentiated or unspecialized, what we mean at that point is that they don't have specific features to carry out a unique job in the body, a unique function. But what they do is they have the potential to develop these features. So stem cells, if we take this example, they've got the capacity to turn into red blood cells to carry oxygen. They've got the capacity to turn into muscle cells to be able to help us move. They've got the capacity to be able to turn into liver cells to help us process food as part of digestion. Okay, and so you can get them from one of two places. There are adult stem cells, and there are embryonic stem cells. Embryonic, because they come from undeveloped babies, fetuses as it were, they have the capacity to develop into any type of cell. Whereas adult stem cells have the capacity to develop into one specific type of cell. So, for example, you can use these to grow organs in artificial environments, like laboratories, for transplants. You often find these, it's important to know where you find them, especially in adults. You can find them in bone marrow tissue. So the tissue inside your bones. That's why you can donate this in order for medical science to be able to use stem cells for functions such as developing artificial organs and things like that. Now, you also find them in plants as well. And in plants, they are referred to as meristem tissue. Good. Right. And then our last area of focus for this is just about transportation within cells. So there are 3 types of transport within cells that you need to know: active transport, diffusion, and osmosis. Now, this table summarises the key aspects of each that you'd be expected to know. So they are all about, just make space for this somewhere, they are all about movement. The movement of particles, molecules. Now, they move different particles in different ways. So, when it comes to diffusion, we are talking about the movement of molecules and ions of any sort, not particularly differentiating. Osmosis is only talking about the movement of water molecules, so it only refers to the movement of water molecules. And active transport, active transport isn't particularly picky either, particles of different substances. Now, when we are looking at diffusion to start with, diffusion is the movement of substances from where there is a high concentration, so where there is lots of this substance, to areas of low concentration, where there is few of the particles. And it is a natural process and it's passive, so it doesn't require any energy. So it might be, for example, substances that diffuse from within your small intestine, nutrients and food that you take on board into your bloodstream in order to be carried around the body. Osmosis is the exact same process, so we're talking about moving from areas of a high concentration where there's lots to areas of particles where there is a low concentration. So again, it is also a passive process, so it doesn't require energy. But only involves water. That's the important difference. Now, as well as that, the water has to move across a partially permeable membrane. So that's a membrane around a cell that contains holes in it that only let specific things through. The final thing is active transport. Now, active transport, you've got to think about, is the opposite. So it's moving substances from where there is a low concentration to where there is a high concentration of it. Now, because it's different, this process requires energy. So an example of where you might find it is the absorbing of minerals from soil to plant roots. Good. And so we've gone over first bit of the specification on cell biology. So watch the video, take notes as you need, and then have a go at the Educate quiz that you've been sent out alongside this, and hopefully it'll help you recap this content for revision.